Hi, my name is Rachael Aubin, and I am a fourth-year PhD student in the Camara Lab at the University of Pennsylvania. Today I'll be talking to you about ConDecon, a clustering-independent method for estimating cell abundances in bulk tissue using single-cell RNA-seq data. Biological tissues are complex cellular systems composed of millions of cells grouped in hundreds of cell types that interact to produce biological function. As a result, characterizing the cellular composition and heterogeneity of these tissues is fundamental to understanding the relationship between cell phenotype and tissue function, and has been a major area of study in biology and medicine for over a century. Recent advances in single-cell RNA sequencing has revolutionized the study of tissue composition by enabling the characterization of cells without the need for predefined markers. However, to use single-cell technology to establish associations between cell composition and other variables, like clinical data, requires generating and analyzing large cohorts of single-cell RNA-seq data, which is often technically, computationally, and financially difficult, especially compared to bulk RNA-seq methods. Moreover, there already exists large repositories of publicly available bulk data. This has led to the development of computational methods that use single-cell data as a reference to predict cell type proportions associated with bulk data. These approaches are often referred to as gene expression deconvolution. Current methods for gene expression deconvolution use matrix regression such as support vector regression or non-negative least squares regression to represent the overall gene expression profile of a tissue, represented as G in this example, as the linear combination of cell type specific gene signatures, represented as S. These gene expression signatures are inferred from the reference single-cell data by methods like clustering and differential expression analysis. As a result, these approaches work well when the cell types of interest are well characterized and cluster distinctly. However, poorly characterized cell states or continuous cell processes like differentiation cannot be easily discretized into cell populations. These nuanced cell populations are often overlooked by clustering-based methods. Therefore, there is a need for clustering-independent approaches that take full advantage of the reference single-cell RNA-seq data to infer single-cell abundances rather than cell type abundances in bulk tissue. Here, we present a continuous deconvolution method named ConDecon for inferring cell abundance from gene expression data of bulk tissue without relying on cluster labels or cell type specific gene signatures at any step. ConDecon is a clustering-independent machine learning approach that uses the gene expression count matrix and latent space of the reference single-cell data to estimate the cell abundance of each bulk sample. The aim of ConDecon is to predict a probability distribution across the single-cell data that represents the likelihood that each cell in the reference dataset is present in the query bulk dataset. In order to make inferences about the cell composition of bulk tissue, ConDecon assumes that the correlation between the bulk and single-cell data can be used as a proxy for inferring the probability distribution associated with cell abundance. For that purpose, ConDecon uses the reference single-cell data to generate a training dataset that contains paired bulk and cell abundance profiles. Simulated cell abundance distributions are modeled as a Gaussian mixture model across the single-cell gene expression space and aggregated to form simulated bulk transcriptomic profiles. We then take the correlation between the simulated bulk and real single-cell data to find the distribution of correlation values. We do this over and over to generate a large and heterogeneous training set that is based on just a handful of single-cell experiments. We would now like to find a relationship between the simulated cell abundance distributions and the correlation distributions. To do this, we use the latent space of the single-cell data to reduce the dimensionality of these two distributions. More specifically, we use the latent space to expand the distribution of cell abundances and correlations on a basis of functions. This linear expansion reduces computational complexity and allows us to estimate the probability of every cell in the reference single-cell data without overfitting our model. Finally, we find a model, h of x, that maps between the space of correlation distributions, x, and the space of cell abundance distributions, y. With our trained model, ConDecon can use the similarity between the reference and query data to predict the cell abundance profile of every bulk sample. As proof of concept that ConDecon can truly infer changes in cell state, we applied it to simulated bulk and single-cell RNA-seq data for which ground truth cell abundances are known. For this, we used the software Splatter to simulate single-cell RNA-seq data containing continuous cellular processes, such as differentiation or immune cell activation. These simulations represent scenarios in which clustering-based gene expression deconvolution methods might struggle. In this analysis, we focus on cell differentiation trajectories with 3 different topologies and 13 levels of differential gene expression. As you can see from the 3 examples, each trajectory consists of a precursor state, colored in dark purple, and 2 or 3 terminal states, colored in yellow. For each simulation, we also generated 25 unique bulk gene expression profiles by aggregating cells based on a Gaussian kernel centered at a random point along pseudotime. I want to draw your attention to the visualization on the left side. In figure A, B, and C, I am showing a UMAP representation of 3 different topology trajectories colored by pseudotime. Then for each trajectory in the top row, I am showing the true cell probabilities used to create 3 different synthetic bulk profiles. What I find really exciting is in the bottom row where I'm showing ConDecon's predicted cell abundance for those same bulk samples. Overall, we find that ConDecon's estimated cell probabilities are strongly correlated with the simulated probabilities across 39 different trajectories. These results demonstrate the utility of our clustering-independent approach for gene expression deconvolution. We then sought to benchmark ConDecon against other deconvolution methods. To do this, we aggregated ConDecon's inferred cell probabilities into cell type abundances to match the output of current methods. We then used a published benchmarking pipeline that evaluates the accuracy and stability of each method to increasing levels of noise in the reference single-cell data. In this case, noise was introduced by removing either a small, medium, or large cell population from the single-cell data while preserving that cell population in the query bulk data. We then took the Pearson correlation between the estimated and true cell type proportions. We find that on average across 6 different datasets, ConDecon outperforms 16 of the 17 other methods. In addition, ConDecon is particularly robust against situations where a large cell type was removed from the reference. To further benchmark ConDecon, we leveraged 2 bulk RNA-seq datasets with paired ground truth cell type proportions determined by FACS. We find that ConDecon's average cell type performance consistently outperforms other methods when compared with FACS data. Overall, these results demonstrate that ConDecon's cell probabilities can be aggregated into discrete cell type abundances to produce comparable predictions to those of clustering-based methods. Next, we used ConDecon to estimate dynamic cell abundances associated with well-characterized biological systems. For this, we used the Tabla-Muris bone marrow dataset, which consists of single-cell and bulk RNA-seq data from mice ages 1 month to 27 months. This is an ideal system to study continuous changes in cell state associated with age-related processes. For this analysis, we focused on known changes in B cell abundance that occur during mouse development and maturation. We find that ConDecon is able to recapitulate consistent shifts from an abundance of pro and precursor B cells in young mice, less than or equal to 3 months, to an abundance of naive B cells in mature mice. These results are quantified in the box plots on the right, where we find a significant fold change in each B cell population between young and mature mice. We compared these results with other methods and find that ConDecon consistently predicts a significant fold change in the abundance of each B cell population between the young and mature mice. Finally, we sought to investigate the relationship between tumor-infiltrating microglia and the mesenchymal transformation of pediatric ependymoma, a rare and aggressive brain cancer in children. To that end, we applied ConDecon to a previously characterized transcriptomic atlas of these tumors consisting of bulk and single nucleus RNA-seq data. Consistent with clustering-based deconvolution methods, ConDecon predicts that the cell type composition of ependymal tumors is quite heterogeneous. To better understand these dynamics, we then defined a mesenchymal and microglia activation score for each tumor. These scores represent the relative density of cell abundance inferred by ConDecon along the EMT-like or microglia trajectory, respectively. When we look at the relationship between these two scores, we find that there is a strong correlation between the mesenchymal and microglia activation score. These results suggest that the mesenchymal transformation of ependymoma tumor cells is paralleled by the activation of tumor-infiltrating microglia, consistent with the central role of tumor microenvironment in this process. In summary, we propose a new method called ConDecon, a clustering independent deconvolution algorithm for inferring single-cell abundance distributions. We find that ConDecon's estimation of discrete cell type proportions is comparable or superior to current clustering-based methods. In addition, ConDecon is able to recapitulate continuous changes in cell state associated with simulated data and well-characterized biological systems. Finally, we use ConDecon to look at the relationship between the mesenchymal transformation of ependymoma tumor cells and the activation of microglia. Taken together, our results demonstrate that an unsupervised, clustering-independent approach, like ConDecon, can elucidate previously undetected cellular processes in bulk tissue. With that, I would like to thank everybody in the Camara Lab, especially Javier Montalongo, as well as my thesis committee. For any questions in this project, please feel free to reach out via email. Thank you.